You are welcome to class. This is CBG 211. We want to talk about cell biology. And today we are looking at cellular respiration. And under this cellular respiration,
You are welcome to class.
we are going to have
CBG 211.
we are going to look at some fundamentals aspects of cellular respiration. We're going to look at glycolysis. We are going to look at the Krebs cycle, and we're also going to look at the electron transport chain. So those are the three major aspects that we are going to consider under the electron transport chain, under the cellular respiration.
You are welcome to class. This is CBG 211. We want to talk about cell biology. And today we are looking at cellular respiration. And under this cellular respiration, you are welcome to class. We are going to have CBG 211. We are going to look at some fundamentals aspects of cellular respiration. We're going to look at glycolysis. We're going to look at the cycle, and we're also going to look at the electron transport chain. Okay, so those are the three major aspects that we are going to consider under the electron transport chain, under the cellular respiration.
Okay, so let's go. When we talk about cellular respiration, it is a set of metabolic reaction and processes that mainly occur in organisms. And what they do is that they convert biochemical energy that are being trapped in nutrients into adenosine triphosphate. That's ATP. And ATP is being known as the currency of the cell.
Okay, so let's go. When we talk about cellular respiration, it is a set of metabolic reaction and processes that mainly occur in organisms. And what they do is that they convert biochemical energy that are being trapped in nutrients into adenosine triphosphate. That's ATP. And ATP is being known as
Okay, so plants most of the time produces oxygen, and this oxygen is utilized by the animals, and the animals break down the sugar molecules in them, and the animal releases carbon dioxide. The plant takes up the carbon dioxide, and the plant uses the carbon to make sugar. So we can see this in this equation below, where we have carbon sugar, and then being added to oxygen, and then CO2 is being produced. This is referred to the process whereby ATP is being produced. They are heterotrophs organisms that actually use this pathway. We also have the autotrophs, where they convert carbon dioxide plus water and energy to give glucose and oxygen.
Processes that mainly occur in organisms, and what you do is that
Okay, so plants most of the time produces oxygen, and this oxygen is utilized by the animals, and the animals break down the sugar molecules in them, and the animal releases carbon dioxide. The plant takes up the carbon dioxide, and the plant uses the carbon to make sugar. So we can see this in this equation below, where we have carbon sugar and added to oxygen, and the CO2 is being produced. This is referred to the process whereby ATP is being produced. They are heterotrophs organisms that actually use this pathway. We also have the autotrophs, where they convert carbon to give glucose. Okay, so under stage one, we want to look at glycolysis, a process whereby glucose is being broken down through series of reaction to the production of pyruvate, and then there's a concomitant release of ATP, and thereafter the ATP is being also utilized by the organism.
Oxygen, and this oxygen is utilized by the animals, and the animals bring down sugar molecules in them, and the animal uses carbon dioxide. The plant picks up the carbon dioxide, and the plant uses the carbon to make sugar. So oxygen Okay, so under stage one, we want to look at glycolysis, a process whereby glucose is being broken down through series of reaction to the production of pyruvate, and then there's a concomitant release of ATP, and thereafter the ATP is being also utilized by the body.
Okay, so the glucose gets broken down to glucose 6-phosphate, to fructose 6-phosphate, to fructose 1,6 bisphosphate, and then glucose 1,6 bisphosphate is being broken down to both glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The enzymes that are involved from glucose to glucose 6-phosphate is exokinase. We have glucokinase in liver and pancreatic beta cells, and this part is regulated by activation by insulin and by glucose 6-phosphate or glucagon.
Process whereby glucose is being broken down through series of reactions to the production of ATP.
Okay, so the glucose goes to glucose 6-phosphate,
down to glucose 6-phosphate, to fructose 6-phosphate, to fructose 1,6 bisphosphate, and then glucose 1,6 bisphosphate is being broken down to both glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The enzymes that are involved from glucose to glucose 6-phosphate is exokinase. We have glucokinase in liver and pancreatic beta cells, and this part is regulated by activation by insulin of glucose.
Okay, so like I said, the enzymes involve glucose to glucose 6-phosphate. We have exokinase, glucose 6-phosphate to fructose 6-phosphate. We have glucose isomerase from glucose 6-phosphate to fructose 1,6 bisphosphate. We have phosphofructose kinase 1. And then from fructose 6 bisphosphate to glyceraldehyde 3-phosphate to dihydroxyacetone phosphate, we have adolis.
The glucose goes to glucose 6-phosphate, down to glucose 6-phosphate, to fructose 6-phosphate, to fructose 1,6 bisphosphate, and then glucose 1,6 bisphosphate is put glyceraldehyde 3-phosphate and dihydroxy
Okay. I don't know whether the audio is better now. I realize that the line is breaking. So I want to take this again. I was explaining the breakdown of glucose, from glucose to glucose phosphate, from glucose phosphate to fructose bisphosphate, and fructose 1,6 bisphosphate. It's been broken down to glyceraldehyde 3 phosphate and the dihydroxyacid exokinates that converts glucose to glucose phosphate. In liver and pancreatic beta cells, we have the glucokinase, and then we have insulin, which have as an activator in regulating this particular pathway. We also have glucose phosphate and glucagon that also negatively regulate this pathway by inhibiting of insulin actually activates or favors the conversion of glucose to glucose phosphate. And it's also been noted that ATP is being used in this particular step. So moving forward, we have glucose phosphate going to phosphoglucose isomerase is the enzyme converting to fructose phosphate. We also have that particular reaction is a reversible reaction. That should be noted. So we also have So glucose 1,6-bisphosphate being converted to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This interconversion is catalyzed by triose phosphate isomerase. This can be converted to this, and this can be converted to this. So at the end of this particular reaction, the phosphate of glycolysis is being concluded, and it is referred to as the end of investment or the preparatory phase. So we have two major phases in glycolysis. We have the investment phase and the preparatory phase. So this actually is the end of preparatory phase. Glucose to glucose phosphate to glucose phosphate to fructose 1,6-bisphosphate, then two through aldolase to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So majorly glyceraldehyde 3-phosphate is the one that continues the journey to the next phase, which is the energy generation phase or the payoff phase. So at this particular phase, the glyceraldehyde 3-phosphate is the one that begins this journey, and it moves to 1,3-diphosphogycerate, and we also have 1-phosphoglycerate, which is also converted to 2-phosphoglycerate, and 2-phosphoglycerate is converted to phosphoenol pyruvate, and pyruvate marks the end. Okay, so looking at that from glyceraldehyde 3-phosphate to 1,3-phosphoglycerate to 3-phosphoglycerate to phosphoglycerate to phosphoenol pyruvate and pyruvate. And the enzymes that are involved from glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate is glyceraldehyde 3-phosphodehydrogenase. The enzyme involved in 1,3-bisphosphoglycerate is phosphoglycerate kinase. The enzyme involved in 1,3-phosphoglycerate to 2-phosphoglycerate is phosphoglycerate mutase. And the enzyme involved from 2-phosphoglycerate to phosphoenol pyruvate is enolase. And the enzyme involved in phosphoenol pyruvate to pyruvate is pyruvate kinase. So, and this pyruvate marks the much end at this point where glycolysis ends. So there is a possibility of different fate of glycolysis in the presence of oxygen and also in the absence of oxygen. In the absence of oxygen, one of the possibilities is that lactate dehydrogenase converts pyruvate to lactate. And this happens mainly when you have heavy exercise or happens in the red blood cell. Also, we have in yeast and some bacteria, we have alcohol dehydrogenase and pyruvate decarboxylate can convert pyruvate to ethanol. This is referred to as alcohol fermentation. Whereas in the presence of oxygen, the fate of pyruvate is different. The journey proceeds to acetyl-CoA through the presence of a pyruvate dehydrogenase complex that's engaged in the presence of oxygen, NAD being reduced to NADH, and CO2 being reduced. You have acetyl-CoA. And this particular reaction will actually lead into another set of pathway that we are also going to consider. So the journey continues along that way as glucose is being broken down to pyruvate through the payoff and also through the preparatory phase and the payoff phase, ending at pyruvate, and then the fate of pyruvate being determined in the presence or absence of oxygen, and also depending on the organism or the cell, this is actually happening. If you have a quick recap of what we have been saying about glycolysis. So we have glucose coming to glucose 6-phosphate, glucose 6-phosphate going to fructose 6-phosphate, okay? And then from fructose 6-phosphate to fructose 1,6-bisphosphate that has two fates. The first fate is that it could go to dihydroxyacetone phosphate, or it could also go to glyceraldehyde 3-phosphate, and dihydroacetone phosphate and glyceraldehyde 3-phosphate has possibility of interconversion in the presence of this enzyme triphosphate isomerase. So triacetyl 3-phosphate continues the journey and it moves to 1,3-bisphosphoglycerate, and from 1,3-bisphosphoglycerate to 3-phosphoglycerate, from 3-phosphoglycerate to 2-phosphoglycerate, and from 2-phosphoglycerate you move to phosphoenol pyruvate, and from phosphoenol pyruvate we move to pyruvate. So that is the rundown from glucose through glucose 6-phosphate and fructose 6-phosphate and fructose 1,6-bisphosphate, glyceraldehyde 3-phosphate, 1,3-bisphosphoglycerate, and 3-phosphoglycerate, 2-phosphoglycerate, and phosphoenol pyruvate down to pyruvate. So at the end of this, the net production of ATP that is produced here, we have 2 ATP that is produced here. And so, like I said, in the absence of oxygen in an anaerobic reaction, so the possibility of pyruvate being converted to a lactate or lactic acid is there. And also the possibility of also producing alcohol in the absence of oxygen, which is referred to as fermentation, is there. Now, in the presence of oxygen, the Krebs cycle will actually take place. So having said that, there's what we call the Warburg effect. In this Warburg effect, it means that glucose normally after several reactions can go to pyruvate, and then that pyruvate normally is supposed to proceed through acetyl-CoA to the Krebs cycle on the oxidative phosphorylation. But in this particular case, in the presence of oxygen, we see that pyruvate through lactate dehydrogenase can actually be converted to lactate, thereby circumventing the normal pathway of moving straight to Krebs cycle and oxidative phosphorylation. So what happens here is that there is a bypass of pathway that leads to massive production of ATP. And this is majorly found in cancerous cells. So because the rate of proliferation of cancer is high, and so there is the need to have these cells proliferate at a high rate. So they need much of ATP. So at this point, glucose breakdown does not follow through the pathway. But what happens is that it goes straight through lactate dehydrogenase, converting pyruvate to lactate, and then we have almost 10 to 100 times more ATP being produced. And what happen, it will cause of the cells to be energized, and so they can continue their proliferation. From this illustration, you can see much ATP is being produced, which favors a lot of cells being proliferated. So you now have quite a number of cells being proliferated. Why? Because we have a lot of energy currency in form of ATP that is readily available for this group of cells. And of course, they can now lead to malignancy because a lot of these cells are being produced, a lot of ATP is being annexed, and this ATP is annexed following the glucose that has been broken down to pyruvate and that did not follow the pathway of going through the acetyl-CoA to the Krebs cycle to the oxidative phosphorylation, but bypassing through lactate dehydrogenase in the presence of oxygen, breaking pyruvate to lactate, whereby causing generation of a whole lot of ATP made available for the cells. And the cells will take advantage of this, and then they will use it to continue to proliferate. Now, having said that, okay, starting from this second part of the breakdown of glucose that we said, that the glyceride 3-phosphate going down to 1,3-bis-phosphoglycerate and going down to 3-phosphoglycerate and going down to 2-phosphoglycerate and being broken down for phosphoenol pyruvate and to pyruvate. I told us that you have different possibilities depending on the availability or the absence of oxygen. In the absence of oxygen, it could go to lactate and in the presence of the enzyme lactate dehydrogenase, while in the also it could also go to ethanol in the absence of oxygen, where we have alcohol dehydrogenase and pyruvate decarboxylase. We are producing the ethanol, especially in yeast and some forms of bacteria. Now, in the presence of oxygen, the journey continues. The conversion of pyruvate to acetyl-CoA, that is pyruvate dehydrogenase complex. So the pyruvate dehydrogenase complex facilitates the conversion of pyruvate to acetyl-CoA with the release of CO2 and then NAD being reduced to NADH. So in this particular place, the journey now moved from acetyl-CoA to the Krebs cycle. So this is a process of energy production in form of ATP. And so when this happens, so we can now move to the next one, which is the Krebs cycle. Okay, so what happens in the Krebs cycle or the citric acid cycle? The pyruvate that is coming from being converted to acetyl-CoA, that is converted, acetyl-CoA combines with oxaloacetate to form isocitrate, and then isocitrate to oxoglutarate, to succinyl-CoA, to succinate, to fumarate, and then to malate. Okay, these occur if oxygen is present and the cells in the mitochondria, if oxygen is present in the cell of the mitochondria. So in this stage of cellular respiration, the oxidation of the glucose of CO2 is completed. And part of the reason why we actually exhale carbon 4 dioxide. So at this particular point, the combination of azoacetate with acetyl-CoA that produce a citrate, you can see that we have a 4 carbon molecule here, and then the pyruvate loses a carbon to CO2 and uses the hydrogen ion to form the NADH and become acetyl-CoA. That's from the pyruvate to this place. Before the combination of this, and then we have formation of citrate, isocitrate, alpha ketoglutarate, and the cycle goes on until it comes back to oxaloacetate. Okay, so here we have the release of 2 CO2, the reduction of NADH to NAD to 2 NADH, and then 2 acetyl-CoA that actually entered. Now here you can have a broader picture of it. So at the end of the Krebs cycle, we have per glucose molecule, we have 2 ATP being produced, 6 molecules of carbon 4 oxide, 8 of NADH, 2 of FADH2 that is being produced from this particular cycle that we have that goes round and round. So the aerobics occur in the matrix, that is the inner compartment of it. So this shows the count of the number of carbon that is being reduced or increased. Pyruvate being 3 coming reducing by 1 to form CO2, and then we have 2 carbon acetyl-CoA to form the citrate 6 carbon. We have 6 carbon here. Another reduction of carbon to 5 carbon, CO2 being formed. Another reduction of carbon to CO2 being formed to 4 carbon. 4 carbon still remains, 4 carbon still remain, 4 carbon still remain. Comes down to oxaloacetate that has the same 4 carbon. And then we have a reduction of electron carriers that can also be very, very useful. So along the same pathway, we have that we said from glyceraldehyde down to pyruvate, getting to this point, the acetyl-CoA to pyruvate. So after the Krebs cycle, what next? We now have some of the product that was gotten in the electron in the Krebs cycle moving to the electron transport chain to help in the production of more ATP, because here is where ATP is being produced. So here we have the electron transport chain and also the oxidative phosphorylation. We'll talk about the oxidative phosphorylation is the process in which ATP is formed as a result of the transfer of electrons from NADH or FADH to oxygen by a series of electron carriers. Electron carriers that we have here. So we can see them in complex 1, in complex 3, in complex 4, and also the enzyme here that helps in the conversion to the production of ATP there. So like I said earlier, now this portion where the complexes move the electron from one complex to the other is the electron transport chain, while this latter part that converts the ATP to ATP is the oxidative phosphorylation. At the end of the electron transport chain, we have 34 ATP that is being produced. So here we have, this happens in the mitochondria. This is the inner mitochondria, the mitochondria matrix, and the intermembrane space. Okay, so what happens here is that we have the complexes. The first one, the complex 1, is NADH dehydrogenase. Okay, in which here the ion proton passes through them, and this leads to the carrying of electron from electron transport chain to the next complex. So the next complex, this electron carrier is coenzyme Q. It moves the electron from here from complex 1 to complex 3. So the BC complex to complex 3. Also, after allowing the proton to pass through, the coenzyme C is also electron carrier that moves this from this complex BC to cytochrome C oxidase complex. Cytochrome C oxidase complex is the one that receive the electron carried by coenzyme C. And so the hydrogen, oxygen coming together to form water, and the hydrogen ion also being released. Okay, as it's being released in here, this will eventually cause a particular gradient, and this gradient is the one that's going to make the ATP synthase to be able to produce ATP. So the presence and the movement of this hydrogen ion across the ATP synthase encourages this particular enzyme to convert available ATP to ATP. So this is what happens. So you now have a lot of production of this particular ATP. So at the end of the day, we have glycolysis producing 2 ATP, 2 NADH. The Krebs cycle in total produces 4 ATP, 10 NADH, and 2 FADH, while the electron transport chain produces in total 30 ATP. So in total, making 34 ATP that is being produced. And in some cases, we have a situation whereby electron transport chain produces 34, in addition with the 4 produced by the glycolysis and the Krebs cycle, giving a total of 38 ATP. So this is the overview of that very breakdown from glycolysis down to the electron transport chain. Bacteria may differ in their carriers of electron transport chain, but the number of ATP generated per reduced NADH and FADH is not always a whole number. So some of these things may vary depending on the kind of organism. For every pair of electron transported to the electron transport chain by a molecule of NADH, between 2 and 3 ATPs are generated. So that range, and for each pair of electron transferred by FADH, between 1 and 2 ATP are generated. Okay, so just as that's been explained before, so all this gives the idea of the ATP that is being generated at the end of the breakdown of the glucose. So that is it for the class. Let's have a quick rundown of what we have said. Cellular respiration. We spoke about cellular respiration. That is where we started from. So we said cellular respiration is a set of metabolic reaction and processes that take place in the cell of organism to convert biochemical energy from nutrients to ATP. We also said that a plant utilizes oxygen, utilizes carbon dioxide, produces oxygen, and animal utilizes the oxygen and produces carbon dioxide for plant to produce sugar. Okay. Heterotrophs, the ones that depend on autotrophs, they get the glucose from them, combined with oxygen and bring them to carbon dioxide and water, and ATP is produced. Meanwhile, the autotrophs get the CO2 and get water in the presence of light and convert it to glucose and oxygen. And so I said those that led to how does this glucose being broken down in organism to produce ATP, which is the currency of the cell. The glycolysis occurs in cytoplasm, and it occurs in both prokaryotes and eukaryotes. Okay, so I said at the end of ATP, the net production of ATP produced is 2, because we produce 4 and then 2 is being utilized. So in overview, we said glucose goes to glucose phosphate, glucose phosphate to fructose phosphate, fructose phosphate to fructose 1,6 bisphosphate, and that's broken down to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate moves to 1,3-bisphosphoglycerate. 1,3-phosphoglycerate goes to 3-phosphoglycerate. 3-glucose goes to 2-phosphoglycerate. 3-glucose goes to phosphoenol pyruvate, and phosphoenol pyruvate to pyruvate. It also gave us this overview. Told us that in the presence of oxygen, it moves to acetyl-CoA. In the absence of oxygen, will go to lactate or ethanol. Also told us what we call the Warburg effect. This happens where even though in the presence of oxygen, pyruvate is being converted to lactate in the presence of lactate dehydrogenase, and this encourages production of more times 100 of ATP, which energizes cell, and this favors cancer cells a lot. So I told us that the fate in the presence of oxygen of pyruvate is moving acetyl-CoA and acetyl-CoA entering the Krebs cycle. In this Krebs cycle, what do we have? We have the conversion of acetyl-CoA combined oxaloacetate to isocitrate to alpha ketoglutarate to succinate-CoA to succinate to fumarate to malate and oxaloacetate, and it goes on and on like that. And that will lead to the production of 2 ATP, 2 ATP, 6 CO2, 8 NADH, and 2 FADH2. So I also said that along the same path, the pyruvates also have a fate also by also going from the Krebs cycle into the electron transport chain. And this is where we have the massive production of the ATP, whereby you can see the hydrogen ion crossing into the intermembrane space. So when they cross into the intermembrane space, this as a result of electron being transported from one complex 1 to the other, we have complex 1, complex 3, and complex 4. This creates a gradient that will actually make the ATP synthase to produce ATP, converting ADP to ATP because of the gradient that has been created, and this passing through the ATP synthase of the mitochondria matrix. And this leads to massive production of ATP. So in all, we have a total of 38 ATP that is being produced. Thank you very much for joining. Apologies for the earlier itches in the sound. We'll bring the class to a close.